So I think probably the best way to start this is by you explaining where we are, what is going on here, and then maybe once we understand what it is you're up to, we can delve a little bit more into the science of yeast and how it all works. So please, what is happening here at Berkeley Yeast?
Yeah. Okay. Well, I could talk about this for hours, so I'll try and be concise here. But we're a startup here in the U.S. Bay Area. Um, we've been operating for about 8 years now, and it just all came from the idea that. So I'm. Should I just launch into the whole origin story?
Yeah, sure.
Yeah. Okay. So. I'm a scientist by training. I came to the Bay Area almost 20 years ago now, do a PhD in molecular and cell biology here at Berkeley, and really basic science stuff, computational genetics. And then at the end of that, I wanted to do something a little bit more applied. So I went and I wanted to do something more applied. I want to do something that was kind of good for the world, and I wanted to work with somebody really good. So I ended up taking a position with a scientist named Jay Kiesling. He's a famous sort of academic in the field of engineering microbes. And he was sort of setting the whole field abuzz with applications of engineering yeast to make medicines for curing malaria and all kinds of stuff. And when I was finishing my PhD, his sort of second chapter was around engineering yeast and other microbes to make biofuels. At that time, oil prices were kind of heading through the roof. And I'm thinking, oh, this is going to be amazing. We're going to replace all, fossil fuel transportation fuels, like petroleum-based transportation fuels with biofuels that are carbon neutral. It'll be awesome. Of course, you probably remember oil prices came down with the advent of fracking and all of that stuff.
Yeah.
So, I mean, we worked on those projects, but the unit economics around them were just very difficult. But while I was doing that, I was also making beer in my garage.
Huh.
So, and I would go to just having fun with my buddies, and I'd go to the homebrew store, and I noticed that the most expensive ingredient in my shopping cart was the hops. I was making IPAs, really, flavor forward IPAs. And so the gears start turning, and I'm thinking, well, if I can engineer yeast to make biofuels, surely I should be able to engineer it to make the flavors that ordinarily come from hops. So that was kind of the light bulb moment for me. And so I started, went back to the lab. This is all while I'm, during the day engineering yeast to make biofuels. And, but, nights and weekends started whip up some prototypes, and the prototypes were really encouraging. And so finally this is all under the radar, but finally I kind of work up the courage to tell my mentor, hey, I've got this kind of wacky side project idea. I still remember kind of feeling a little bit, nervous about having the conversation. And he was super excited about the idea, super encouraging. In fact, he suggested, hey, you should go to a brewing conference, start, see if there are people out there that are interested in this. And so I said, okay, and had some great, I did go to an awesome conference called the World Brewing Congress. This is like 8 years ago. Met some incredible people who were, again, very encouraging. And so I kind of had this feeling that I was onto something. I had always thought that I was going to be an academic, but here I am. It's like a pretty cool idea for a startup. So I'm like, okay, I'm just going to write a grant for this small business innovation research. National Science Foundation will fund these projects trying to get kind of science from the bench top out into the real world. And it's a competitive grant. I didn't expect to get it, especially about, I was like, National Science Foundation might, look down their nose at a project around beer. But lucky for me, they funded it, and that was kind of the beginning of the company 8 years ago. All right. So I know I've been kind of...
No.
I warned you.
No, that's good. So just to be clear, the original intention of the company was to be able to brew beer that had aromatic qualities associated with hops,
Yeah.
but didn't need the hops.
Yeah. And so we actually published a really nice research article out of with my old professor. It was published in Nature Communications, and the sort of the culmination of that study was we brewed a beer, actually with a professor of brewing science at UC Davis, a guy named Charlie Bamforth, who I'm sure you've heard of, and the control was just a regular traditional yeast, and the experiment was, this bioengineered yeast to produce some of the, like you said, the aromatics that ordinarily you get from hops. And we did a double-blind tasting over at a brewery called Lagunitas, which is just north, 60 miles north of here. And we got these results back showing that, indeed, we can get yeast to make hop flavor without needing to add hops to the beer. So that was really the sort of first set of prototypes. And now, 8 years later, we still have products that create, yeast strains that create hops flavor. But we have all sorts of different applications as well. So where we're at, to address your first question, what we're doing here is, we try and do what I think of as like an agile development process, which is kind of startup speak. But the agile development process is really an idea from software development where it's like, build a prototype as fast as you can, test it, see what's working, what's not working, iterate, and so forth. And that model doesn't really work that well for, ag tech, right? Which is kind of what I consider our company. However, because yeast is so, relatively easy to bioengineer and it's so fast, and then we can evaluate the prototypes in this pilot brewery. We can engineer the yeast in a month. We can have finished beer in a month, do sensory on it. We can even, we also have a manufacturing plant where we can create liquid slurry that we can get out to brewers all over the country. And with those three pieces, we're able to create products that not only we think are good, but we can actually go vet it out in the real world and get feedback from brewers and their customers.
In a bar.
In a bar, exactly. So it makes it all the way from the bench top to a bar. Yeah.
Right, this is amazing. I want to explore so many parts of what you've just said and learn a little bit more about it. But let's first of all go back to what yeast actually is. Can you explain what yeast is as a microbe, how it behaves, what it likes, what it doesn't like?
Yeah. Oh man, there's so many places to go there. Yeast is a single-celled microorganism, and mostly what it does during the... Okay, so I'll take a quick step back and just talk a little bit about the brewing process. I know all your listeners are already very well-versed, but I like to try and think about it as simply as possible, right? You basically, you start with barley, you soak it in hot water, and a bunch of fermentable sugar comes out. And the reason I specify fermentable is because the yeast is going to ferment it mostly into ethanol. But yeast does just a wide variety of different chemistries in addition to, mostly just converting sugar into ethanol. And probably at least dozens of the chemical compounds that yeast creates will have an impact on the organoleptic properties of the finished the beverage. So I like to think of yeast as just an agent for doing chemistry. You start with wort, the barley water, and it transforms that wort into the things that the just such a wide variety of different liquids that we all love.
Why does it make alcohol? Like what is it about its metabolism that produces alcohol and carbon dioxide and these other chemicals you're talking about?
Wow, that's like a very kind of existential type of question. So there are lots of different species and strains of yeast, like a gazillion in the natural world. And if you were to go out and grab one off of tree bark or off of a rotting fig or something like that, it might not be incredibly adept at making ethanol like the way that the strains that we work with are. But over the centuries or millennia and centuries and decades, humans have basically selected yeast strains that are really good at making ethanol. So they make ethanol in the natural world, but through a process of domestication, not dissimilar from how, wolves are now are the ancestor of dogs, and there's a bunch of different specialized versions of them that look very differently and act very differently and help humans in different ways. Same thing is true for brewer's yeast, and some of them, they've been selected for a variety of different reasons. Oftentimes in some cases it's just like make as much ethanol as possible. But in many cases there is either a trait that makes them easier for a brewer to work with, or, for example, it's really nice that when you're done fermenting a beer, the yeast will settle out to the bottom of the cone, and then you can use that to then go inoculate the next batch of beer. So there's all kinds of crazy
beneficial
traits.
properties to it that might be good for brewing process.
Yeah.
So there's a sort of co-evolution, symbiosis thing there between humans and yeasts that's evolved over the years.
Yeah. And some yeasts don't make ethanol at all, and others are very effective.
And so the purpose do we know? Is it to sort of sanitize its environment? Like why is it shitting out ethanol?
It definitely provides a competitive advantage
against bacteria and other microbes.
Yeah, and a lot of this stuff is, it's sort of like speculative, but scientists speculate that, let's say there's a sugar source and it wants to make sure that it gets to eat, it being the yeast species, wants to consume all of it without any competition from its friends. And its friends all can't tolerate high levels of ethanol, so let's go ahead and just kick out a bunch of ethanol. But there's other reasons as well. So there are different modes of metabolism that are sort of common to a large, range of species in the natural world. There's basically, and this is similar to how. So it can basically use I don't know if you've heard of the Crabtree effect, but basically there are two different ways that yeast can metabolize sugar. There's more than that, but two main ways. One is basically it wants to extract as much energy from the sugars as possible, but it takes longer. But it creates ATP is the sort of energy, the unit of currency.
Energy currency.
Energy currency, exactly. So there's a mode where it says, I'm going to maximize energy currency, and that kicks in most yeast strains when there's a low concentration of sugar available. But when there's a high concentration of sugar available, many of them switch to this mode where it's like, I'm going to consume this fast, create a bunch of ethanol. It's not particularly energy efficient, and maybe it's been selected to do so to outcompete its friends, and then it can sort of continue metabolizing in a more energy efficient manner.
Okay. All right.
Down the rabbit hole. And
how does oxygen factor into this, or lack of oxygen?
Oh, that's a great question. So definitely lack of oxygen. So when I mentioned the more sort of energetically efficient process, that requires oxygen. So another way of driving towards this sort of and roughly speaking, you can call it like aerobic fermentation or fermentative, roughly. And so when you limit oxygen, it drives it towards ethanol formation.
Okay,
for sure.
All right.
Yeah.
And outside of the ethanol formation, you're talking about other compounds that have aromatic qualities, esters and so on.
Yeah.
How do they interplay with the conditions of the fermentation? Lack of oxygen, temperature, time, all of those things.
Absolutely. Yeah. So there are lots of different tools that brewers have at their disposal to influence the finished product. So a great example is if you ferment at a warmer temperature, you're going to get higher ester concentrations, right? So, and then you can also select different yeast strains that might kick off more or less esters. Or so like Belgian yeast strains are more well known for kicking off sort of those like everything from like a bubblegum ester quality to like a banana ester quality. Like the thing that when I drink a Budweiser, the thing that if I were blind drinking it, I would know is it has that nice isoamyl sort of banana character to it.
Yeah.
But then to kind of touch on the tools that we have available to us here where we're engineering yeast to make things, we could take the least ester forward yeast strain, drop an enzyme into its genome, and then have it kicking out esters such that I remember in early days of the company, one of our big things is we like to make strains, make beer, put it in bottles and go take it to brewers and get their feedback. And so one of our first things was we took like a California ale strain, very, very clean, neutral profile, fermented at a relatively normal temperature, 65 Fahrenheit, something like that. And we put this gene that drives more ester production. And I took it to one of our local brewers here and I poured it for him. And he said to me, he's like, this is getting me to completely rethink what I think of a Belgian, yeast strain is, or like what even is a Belgian beer if you can get a California ale yeast to make to make this product. So definitely the tools that we have at our disposal can kind of control those qualities as well.
Okay, so temperature has a factor on ester production. Going back again to sort of the science of this, and putting aside, I guess, different strains, which we'll get onto in a minute. What is it about different temperature that causes the formation of esters or the not formation of esters? Is it something to do with stress on the yeast? How does that work?
Yeah, that's a great question. So yeah, like maybe the default assumption would be maybe these reactions are more favorable at higher temperatures, or like that's from a chemistry standpoint.
Up reaction.
Yeah. But no, I think and again, you might not want to quote me on this, but my I know I'm recording podcast here, but you're literally. But what my belief is that ester metabolism is related to detoxifying stressful conditions. So if you start stressing the yeast by increasing the temperature, the yeast is going to respond by producing more of those enzymes that drive that chemistry, right? So it's going to drive that chemistry because it thinks it needs it to detoxify, but in actual fact, it ends up converting precursors of amino acid biosynthesis and ethanol into those aromatic ester compounds. And then brewers, I think, have sort of taken advantage of that by selecting the ones that, and that's probably where, before all of the domestication that's happened with brewers, that's probably, it was a response to stress. And then probably over the years, brewers got these beers turning out to be, quite, banana ester forward. And then that might have been due to a mutation that arose in the gene that encodes the enzyme that makes that ester. And then they're taking that batch. They say, wow, this is a good batch. I'm going to keep propagating this strain. And that's kind of a good example of how humans have what we do is kind of like genetic modification, but humans have been genetically
the genetic profile already.
That's very much modifying the genetics of the yeast.
You hear that obviously non-GMO food is a big thing, and you hear people talking about that, and you're like, yeah, but we've been selecting for genetics for millennia. We're just selecting it with a bit more precision now. That's the difference.
That's
It's
that resonates with me.
Yeah, I bet it does. So we've been calling it yeast up to this point. Are we talking purely about Saccharomyces cerevisiae, or are there other strains that are relevant here? And do you work with other strains?
Yeah. So Pastorianus is the species name of the lager yeast strain, but we work with ale strains, Cerevisiae, and lager strains, Pastorianus.
What's the difference between those two, sort of broadly speaking?
There's like the genetic history piece, where they came from, and getting back to this domestication process, how they were domesticated and where. But generally speaking, lager beers have been selected to be, much cleaner profile. They've been selected to work over a sort of a different time period. But like the simplest things are, ales are generally fermented at warmer temperatures, and lagers are generally fermented at colder temperatures. And of course, the flavor profiles, lager very clean, and ales like a little bit more hearty, I would say.
Sure.
Yeah.
I sometimes hear people say that one is a top fermenting yeast and the other is a bottom fermenting yeast.
Yeah.
Is there
Well,
any truth
now
to that?
they're ale strain. I'm sure at one point in history, I mean, definitely, there is a wide variation in where yeast is. If you think about where in the vessel the yeast is, whether it's doing its work at the top or in the middle, in the liquid, or at the bottom. But nowadays there are ale strains that will ferment at the top and at the bottom and in the middle. And it also depends a lot on the vessel. It depends on, you might, if you go visit Sierra Nevada, couple hundred miles north of here, they have these flat tanks where you can see the yeast through a window. It's in a clean room, but you can see the yeast, bubbling away at the top of and that's like a traditional way of making lager. But nowadays, yeah, you can see anything from, anything in between. Yeah.
Yeah. Can we talk a little bit about the stages of fermentation? So, and obviously this will vary depending on what yeast you're using and the time and the temperature and the vessel and so on. But just as a sort of process on a timeline, what happens from the moment you take your wort, and perhaps this is relevant to wine as well and any other fermentable substrate, and you pitch yeast in there, and then a few days later you've got alcohol and less sugar.
Yeah.
But what happens in that in between bit in terms of, I don't know, yeast multiplying, temperature changes, production of CO2? Is the production of ethanol linear, or does it peak and trough at different times? Can you kind of break down that process?
Like you said, everything happens pretty darn quickly. If the yeast is happy and healthy, it will ferment usually within, 48 hours. And it depends a little bit about how big the beer is, meaning how much barley you start with and what sort of concentration of fermentable sugar there is. There are lots of other process factors that will affect how long the fermentation takes. But if you're making just like a pale ale, kind of California style pale ale, a lot of that business will happen where the sugar will be consumed within 48 hours, where a lot of that CO2 will evolve within 48 hours. The ethanol will be produced within 48 hours. But then there's a lot of extra work that the yeast will do from that 48-hour mark until, let's just call it, a week later. After about a week, you probably, unless you're doing anything crazy, you probably have a beer that could be a finished beer. Of course, in this area of the country, you get a lot of dry hopping, and that generally speaking adds about a week of additional timing. And there's all sorts of experts you could talk to about sort of the chemistry of dry hopping. But generally, the idea is you add hops to the tank after the yeast is done fermenting. And depending on who you are, you might leave those hops in the tank for 24 hours or, a week. You might recirculate the tank while you're hopping it. So there's a lot of other factors there. And of course, we're really interested in the chemistry of hops.
Not using them at all.
Well, we're actually, I'll just say that the further along this company has come, it's a fun story to say, hey, you could get everything that you get from hops from yeast. But the reality is that hops is a very diverse, it's got a very, like what the things that hops can do to a beer are vary tremendously. Um, Whereas, generally yeast, if we engineer it to make a particular flavor or aroma, it does that exact same thing every single time, which is, kind of a blessing and a curse. One of the things that I love about hops is that you put it into the tank and you don't necessarily know what you're going to get at the end of it. And I see that as, very much an inspiration for the work we do here. And I also think that our yeast is at its best when you're sort of mixing interesting hops with the flavor and aromas that you get off our yeast. That's where I think
Collaborative.
and it's very much like the way that brewers use our yeast is as a tool, similar to how they use hops as a tool or process as a tool. So we're just another sort of tool in the toolbox. Yeah.
Again, I want to dig into that a little bit more in a little while, but just talking about fermentation still.
Yeah.
Obviously yeast eats sugar, but what else does a healthy fermentation need? Are there certain nutrients that need to be present? Are there
Definitely.
conditions under which if fermentation will stall or not work at all?
Yeah. So the biggest thing that comes to mind for me, I mean, the biggest example for me is it must have amino acids. And amino acids require nitrogen, right? So, barley, you can get tons of sugar from barley, but you also have to have a nitrogen source. And the reason you need nitrogen is because proteins are built from, not only, they require nitrogen. So the process of fermentation, in order to have happy, healthy yeast, you need amino acids. And you can either get the amino acids directly from the barley, or the yeast can synthesize other nitrogen sources with the sugars into those amino acids. And then again, not to try and go too far down the rabbit hole, but proteins are they're just strings of amino acids, right? So you have to have all the amino acids to be able to put them together into those strings in order to have enzymes, in order to have life, right? So the reason that example kind of immediately comes to mind is because one of our leading technologies is a diacetyl is related to one of the most persistent off flavors in the brewing world, which is called diacetyl. Everyone knows it's an awful flavor, kind of
buttery,
butter, butterscotch. But that's almost like a euphemism for how it can present. It's kind of like movie theater popcorn butter, but also has a sort of like rancid yogurt quality to it.
Yeah.
Nobody wants it.
Butterscotch
doesn't sound that bad, right?
Yeah,
exactly.
Rancid butterscotch, not so much.
Yeah. So the reason that we sort of have gone so deep into amino acid biosynthesis is because diacetyl actually comes from a branch of yeast amino acid biosynthesis. So yeast is it produces diacetyl kind of like incidentally from a pathway of just making certain amino acids. So what we've done, and again, we've spent so much time on this, which is why it's sort of front of mind for me. But what we've done is we've sort of balanced the metabolism of yeast so it makes enough of these amino acid precursors so it can still make those amino acids, which are essential for its growth, but not make so much of it that you end up with a lot of precursor that can turn into diacetyl. And that's that's a big sort of name of the game for sort of developing our technology. Is a lot of times it's a balancing act. Like, you don't want so much of it that you end up getting off flavors, but you need enough of it. So it's not like you can completely eliminate a part of metabolism. You have to like do this balancing act.
So is diacetyl in part a symptom of like a low nutrition wort, and therefore the yeast is forced to synthesize amino acids to compensate?
Definitely, we believe that the amino acid content of the wort absolutely contributes to the diacetyl potential. There are some other things too, like in U.S. brewing, diacetyl has gotten really famous as a result of dry hopping, because a lot of these, especially, I don't know, 6 to 8 years ago, there was a trend in hops where there was just generally overall less kilning done to try and preserve some of the flavor from the hops. Okay, what happened was, by kilning less, there are all these dextrinase enzymes in the hops, and by kilning at lower temperatures, you ended up with more of those dextrinase enzymes, which means in beers that have appreciable dextrin levels, all of a sudden those dextrinases are kicking off more fermentable sugar. Then yeast is kind of at the end of the kind of like finishing fermentation.
Oh, when you're dry hopping and you're sticking in basically more fermentable sugar into the mix.
Yeah. So the yeast is kind of like, oh, I'm ready to go dormant right now. Like I've done everything I need to do, and then all of a sudden there's some new sugar, and it's like, well, I don't know what to do. Well, there's not enough amino acids around, so like I'm gonna just flood the zone.
Right, and then it pushes out a load of diacetyl.
That's been that's been the bane of many brewers' existence here.
So interesting.
And so the cool thing about the application here is, if you want to kiln your hops at a lower temperature so that you preserve more of those aromatics and not have to worry about diacetyl, well, like that's what
because you've switched it off.
Yeah, exactly.
Right.
Or we've dialed it down to like that perfect zone where it still can make those amino acids, but never get above sort of the sensory threshold. Yeah.
I want to get on to how exactly you go about modifying the genomes of these yeasts.
Yeah.
Is there anything else we should cover before we get there, just to do with fermentation in general and yeast, or do you think we've kind of got there? It seems pretty complete to me, but I just don't want to miss anything before we delve into what you're up to.
Yeah, I mean, the one other thing that I think your listeners would find interesting is that you can also use, and maybe we get into this later, but you can also use bioengineering to reduce the amount of ethanol that the yeast makes in the first place, right? And I think, and one of the things that I think is really interesting about that is you can, the same way, I mean, the way I think about bioengineering is you can, like, if you start with wort and the yeast is doing probably thousands of different reactions and transforming it into beer, and that's because the yeast has thousands of different types of enzymes in it doing those crazy reactions, right? So I think at the outset, I kind of summarized, I oversimplified it, and you kind of brought me back about, what else is required here. There are lots of different reactions that are happening. But the other thing that's really interesting is that, so if most of the reactions are transforming sugars into ethanol, there are so many other reactions that have such a big impact, and nowhere is that more clear than if you use bioengineering to just eliminate the ethanol production. The yeast still has to do tremendous amount of chemistry in order to yield a product that is beer, or in this case, non-alcoholic beer. So it's I'd definitely love to talk more about sort of
so when people say, oh, fermentation just produces alcohol and CO2, it's a gross oversimplification.
Gross oversimplification.
Because there are all these other mechanisms taking place, all this additional biology, chemistry, I don't even know which it is.
Both. Yes, 100% both. Yeah.
That are producing all these flavors that are the things that we actually enjoy from fermentation. Actually, CO2 is maybe something we haven't touched on in depth. What's going on there? Why is carbon dioxide a byproduct of this whole process?
Yeah, I mean, in a nutshell, sugars are composed of carbon and oxygen, and amongst other elements. And as you break those sugars into, break those down, metabolize them, one of the end products is ethanol, and one of the end products is CO2. So it's literally like
It's
For
chemistry.
yeah, exactly. It's part of you could look at the basic chemical reaction and CO2.
Yeah, they're all carbon, oxygen, hydrogen combinations,
Yeah,
right?
exactly,
Yeah.
right? So the sugar is, if you just break it into the chemistry in terms of reactants and products, if sugar is a reactant, CO2 is a product, ethanol is a product.
Yeah, great, clear. Okay, so simple question. How do you go about genetically modifying yeast?
Oh yeah, very simple. Yeah, it is actually it is. I mean, it's pretty simple. I mean, it's complex in some ways, but it's very simple. Oh, where to even begin? So the first thing you need to do is, let's say you want to introduce a new piece of DNA into the genome of yeast, and why don't we actually just unpack what that even means?
Yeah.
So yeast is, a brewer's oversimplification of yeast might be like, it's just a bag of enzymes. Reactants go in and products come out. But actually, yeast is so much more than that, and that's kind of where the biology of it comes in. And it's got lots of different organelles. For example, the nucleus is where the yeast contains all of the DNA, and that's just the code for what enzymes will be produced and what other proteins that have various functional various functions. Like, what are all those things? It's the blueprint. All right. So what bioengineering is it's basically, you can add genes, you can add different genetic material to that blueprint, and there's only one in some organisms there's only one blueprint. In some yeast strains, there's only one blueprint, one genome. In brewer's yeast, there's actually, the standard is 4 copies of the genome. It's called a tetraploid. So one of the challenges for engineering it is, not only do you need to get one, new piece of DNA into it, but you have to get 4 new pieces of DNA in order to make it genetically stable.
So that each cell has 4 genomes.
Exactly.
Why?
Yes.
Why?
That's such an interesting question. Again, I'm going out here, but like, I believe that has more to do with evolution and sort of evolvability than it has to do with anything else. Like, let me put it. Let me give you an example. If you have this is getting really kind of like deep into, not that deep into evolutionary biology, but I'll try and make this kind of straightforward. Is, let's say you have one copy of a genome and something bad happens to one of the genes, like you're toast. Okay.
Yeah.
But if you have 2 copies and then something bad happens to one, you still have one. But then what's even more interesting is that you can not only have something bad happen to one copy, but you can let it evolve into something that does a different function,
Yeah.
right? Or another example that you see in brewing all the time is, you see like genome duplications around, or sorry, in terms of brewer's yeast, you can see duplications of certain genes that might be really important. So one example is, and if you haven't talked to a guy named Kevin Verstreppen, you should chat with him. He has done some really fascinating stuff on the genomic history of different brewer's yeast in particular. And if you go out and look at brewer's yeast versus wine yeast versus yeast that haven't evolved to do fermented beverage process production, you'll see these crazy variations in how they metabolize sugars. Okay, so if you look at a brewer's yeast, maltose is the primary sugar that yeast is converting into CO2 and ethanol, what have you. It also has glucose and maltodextrose, a few other important fermentable sugars, but mostly maltose. Whereas if you're doing a wine fermentation, there's not much maltose there. Okay. Now, if you look in the genome of the brewer's yeast versus the genome of the wine yeast, you see incredible numbers of genes that encode the machinery required to bring maltose and metabolize maltose. So probably a piece of why yeast genomes are so tolerant to having multiple copies, and quite frankly, like tolerant to any number of different kind of modifications, why they're so robust, is because that allows them to kind of evolve new traits. And in the context of brewing, it's like, oh well, there's way more maltose in this wort that these brewers keep feeding me. But you can imagine out in the wild, there's all sorts of different insults that it needs to evolve and respond to. Okay, so that's
it's like insurance policy and an R&D department all in one,
right?
or in four.
Yeah, absolutely.
Yeah.
So back to the question of how we mostly what we do is we either insert new DNA into the genome of yeast or we delete DNA from the genome of yeast. And speaking of just amazing sort of molecular machinery that these yeast have, if you can figure out a way to cut the genome of the yeast, it figures out how to repair it, right? And if you also supply it with a piece of DNA that looks kind of like one broken end and another broken end, then it will use that new piece of DNA to repair itself and put something new in there, right? It's the fact that this exists is, I mean, it's pretty stupendous. It like gets at exactly what we're talking about. Like yeast are, and organisms in general are just like highly dynamic,
Adaptable.
very adaptable. Their genomes are always changing, even though we, I think tend to think of genomes as static. They're incredibly mutable and incredibly fast moving. And nowhere is that more apparent than if you look at the history of, how yeast have evolved to ferment do different things in the natural world.
So when you're talking about introducing new bits of DNA, genetic code, like we've all seen kind of computer generated graphics of what DNA looks like, the helix and everything.
Yeah.
But how does that actually work in the physical world? Because we're talking about very, very small bits of data here. So you're not using a tweezers to do this, right?
No, that's exactly right. It is a molecular. It's effectively like a molecular tweezers. Molecular scissors is cliched in the sort of bioengineering landscape. But basically, there are proteins and proteins. They're just as like. Just trying to think about in a sense of scale. Like we're talking about, a sense of scale that is obviously not visible to the human eye. Like you can't see yeast, and yeast have a gazillion proteins inside of them, and these, a gazillion nucleotides that form these genomes. So it's hard to even like
comprehend the scale.
Yeah, exactly. Yeah. But when you get down to that molecular level, you have proteins that will recognize a specific, series of nucleotides and make a cut specifically. And that
a nucleotide is a chain of proteins?
I'm sorry. Yeah. Okay, here we go. So DNA is composed of nucleotides, the A's, C, T's, and G's of, and then proteins are composed of amino acids. And amino acids there are 20 different amino acids. And the DNA with the nucleotides encode genes, and the genes are read, again, by these little molecular machines encoded by proteins, and these proteins actually will scan along the DNA and convert those nucleotides into a protein. Like that, and I'm skipping a few steps, but like that's like the central dogma of molecular biology. It is a wonder.
It sounds like science fiction.
It's great. It's so science fiction. The natural world, you go, the deeper you go, the more science fiction.
Yeah, I mean, it's like with physics and quantum theory and all. The more you understand about science, the more it sounds totally fantastical.
Yeah, I couldn't agree more. The more you know, the crazier it gets. So there are, these other enzymes or proteins that will recognize specific nucleotides in the genome, cut them, and then you can basically mix in DNA that you want to insert into the genome, and it finds, though, by virtue of the broken ends looking similar to the piece that you introduce, it will naturally repair itself and introduce the.
So you're using biology as the scissors.
100%.
Yeah.
Yeah.
Okay.
It's, So that the next to kind of step backwards, you may wonder, how did you make that piece? Okay, so on that sort of line of thinking, why does yeast do that? Well, I mean, I think it's safe bet to say that it by having the ability to repair itself and having the ability to introduce new DNA into its genome, it's more adaptive. Okay, so here's something I find really crazy. The way that we can encourage the yeast to take up new DNA is basically by stressing it out. And I figure this is like in the natural world, when a yeast is stressed, it's thinking like, I need to try some stuff.
Yeah.
Right?
Fight or flight kind of mode.
Yeah.
That's I'll give anything a go at this
Yeah,
point.
exactly. And so what we do when we insert, I want to talk a little bit about how we make that new piece of DNA in the first place. But what we do is we stress the yeast out, we get them to cut their genome, and then by virtue of being stressed out, it will suck up all of this exogenous DNA that we are supplying, and then the DNA will find. And the other thing that I just, again, sense of scale, we will that process involves us offering this repair DNA to, again, a gazillion yeast cells, and we only need one of them to repair itself in the way that we envision in order to kind of get what we need. Right? So that's sort of the mechanism by which we introduce the new DNA. Now to get back to the question of, well, how do you get that piece of DNA that you want to insert in the first place? And that's all crazy molecular cloning stuff, but in that room, there's a bunch of, it relies heavily on a technology called PCR, polymerase chain reaction, which is another sort of modern marvel of biotechnology. But basically what you can do is you can synthesize a piece of DNA, and then you can amplify it, and then you can put it into what's called a vector, which basically is a piece of DNA that you can start shuttling it inside and out of other microbes. Like E. coli is really good at amplifying pieces of DNA. So these are kind of the tools of molecular biology, and I don't want to bore your listeners too much on this note.
So the purpose of that is to observe what happens in the real world when you shuttle this bit of DNA in and out. You can kind of test it.
It's actually more like a factory for building pieces of DNA.
Okay.
Right? Because the new pieces of DNA that we're inserting in the genome generally they never existed in the natural world. So how do we kind of recombine and
how do you know what DNA to make?
Oh man, this is such a good question. So this is a field this is like the field of metabolic engineering in a nutshell. It's over the last many decades, as we've characterized DNA and the gene sequences and the related sort of molecular biology elements that are related to genes, like we've continuously sort of improved our understanding of what various different genes do, what control elements do, and I mean, so over the decades, we know more, and like we basically imagine it as though the natural world has the biggest library of DNA that you could possibly imagine. What we have done over the decades is sort of like checked out those books and read them and figured out what they're about, and then put it into a barcoding system and said like, okay, we know this and that about this piece of DNA.
And you know it by switching it on and off and seeing how it modifies the organism.
That is the 100% the way that it was done in the early days. And that was like, when you think about old school genetics, folks that were kind of pioneering this in, the yeast genetics back in, I don't know, 70s, 80s, 90s, like before there was even, and again, so yeast, not only is it incredible for making beer, making wine, all that stuff, but it's also an incredible model system for understanding just biology in general. Why is it such a great model system? Well, it's very like genetically tractable. It's easy to like. Has a relative. Oh man, I'm going so far down a rabbit hole, but I'll just keep going.
It's all good.
Just keep going.
Yeah.
So it's got a relatively compact genome, and it replicates quickly, so that makes it easy for scientists.
You can work fast with it.
Yeah, you can work fast with it. So
if you try doing it with an oak tree,
that's gonna exactly, or humans for that matter. It's pretty tough. Yeah. So it's a great model system for understanding humans. And in some of the, before we even had like a yeast genome, and this is before we had a human genome, like a yeast genome was sequenced before the human genome was sequenced. But before we even had a yeast genome, what scientists were doing were characterizing these genetic elements through, let's delete something or let's mutagenize it or whatever, but like let's eliminate that thing and see what it doesn't do as well, right? So sugar metabolism, for example, you want to see, it's funny, if you look at the gene names for maltose consumption, they're all called MAL1, MAL2, MAL3, and lots of different clusters of genes are named according to this convention. And the reason why is because like 50 years ago, somebody was saying, well, I'm going to mutagenize this yeast, and oh look, now it doesn't consume maltose properly, so I'm going to call that MAL1. Like that, and then over the decades, people were like, oh, I actually know what sequence what gene was deleted in order to, and now we know like all the A's, T's, and C's and G's that make up that gene. So that was like library barcoding, 50 years ago, and it's just gotten progressively more and more impressive. We also know a lot about, like, you can characterize the proteins through chemistry, through sort of like, that's kind of the field of biochemistry. And then the field of bioengineering, really, it's like, we all kind of, it's like, it all relies on all of this basic science that's been done over the years. So now I know, let's just take the example I was using before of these yeast strains that prevent diacetyl precursor production. There's an enzyme called ALDC, alpha-ketolactate decarboxylase. ALDC is the acronym. So we know exactly what chemistry, we know exactly what that gene is, the A's, T's, C's, and G's. We know, and it codes for certain amino acids that fold into a protein, and we know that protein takes, we know what reactants it takes and what products it produces. Okay. So then you can take that piece of DNA and insert it into the genome of the yeast. Okay. And that's the whole thing. It's pretty simple.
It's like Lego bricks, right? You're just taking one, I want a yellow one in there, and you're and the yeast is just going, cool, I'll have that. Yep, I'll take that.
Paste.
Copy paste. That's definitely an oversimplification. But so broadly speaking, you're taking Lego bricks and you're using biology itself in order to snip these bits out, and then the yeast, by its very nature, is sort of accepting of this code.
Yeah, exactly.
Wow. Is there I mean, this is maybe going even further down the rabbit hole, but my knowledge of biology is fairly limited, but are genes always sort of working in isolation, or is there sort of knock-on effects of you take one out, switch one off, or bring one in, and then it modifies the way that other genes behave?
100%.
How we go. Maybe it's not as limited as I thought, my knowledge.
No. That is, it is a complex, it's a web.
Yeah.
And yeah, there's a lot there.
So you can't just copy paste. You have to be sort of cognizant of other genes and how that one's gonna, and then now we need to pull that one out or put something else in.
You have great intuition for this. Yeah. And honestly, I would say that is one of the things, and I'm trying not to sort of like, turn this into a story. I'm loving the conversation. I'm not trying to kind of like pitch our products or anything like that. But what I will say is that the thing that I believe makes the work that we're doing so special is when you introduce when you're copying and pasting something in, that might create like a bug. Like if we think about it as like software code,
Yeah.
that might create a bug, like a knock-on effect that presents as a bug. For example, when we made some of these low diacetyl strains in the early days, and we weren't thinking about kind of what those knock-ons effect, or we didn't understand what they were initially, we would put the enzyme or put the gene for the enzyme in, and it would be like, oh, there's no more diacetyl, like everything's going great. But then we, in the hands of some of our brewers or customers, they would say, well, it did great, but actually, its viability is not great after a couple rounds of ferment, or sometimes, like, generally it would be like a couple rounds of fermentation. We say, okay, that sounds like a bug that we ought to fix. So sort of circle back to what we were talking about earlier, where we were thinking, oh, okay, we want to make sure that this enzyme isn't cranking so hard that it's actually robbing the yeast metabolism of those basic amino acids that it needs just to survive. So it's like, okay, I copy pasted the wrong thing. Let me go back to my library of things that I can copy and paste in. I'll paste something different, and through that sort of like, now it's like, okay, I don't think this is going to have that viability issue or that repitching viability issue anymore. Get it back to the customers. They're like, oh no, this is actually working perfect for me. I can repitch this ad infinitum. So it is very much a process of copy paste. Yes, there are knock-on effects. Let's tweak and debug and get to a place where all of the important things that, every trait that's important to a brewer is met by that new design.
So there's a degree of trial and error in it. But of course, every time you make an error and try something new and then fix the error, you've learned something.
Yeah.
And then the trial and error becomes more precise as time goes on.
100%. Yeah.
Yeah. Okay.
Yeah.
I mean, it's just incredible. Like, it's so remarkable that we have this sort of power to be able to modify these things and create flavor through biology.
It's amazing.
And manipulate flavor in that way.
You work on this every day. I'm working on this every day for almost the last 10 years. And most days when I come in, it feels very unremarkable. But the first time you insert a piece of DNA and you have the parent and you have the newly created strain, and you ferment this one, and it's like, one outcome, and this one another like a completely different flavor and aroma. It's like mind-boggling.
It really is, yeah.
We can pour a few samples that are illustrative of that.
Yeah.
And I mean, I think every time I do a demo for a customer or for someone that's new to the company, you kind of see that, and it is really inspiring, because working on a startup for years and years and years can sometimes feel a bit like a slog, but when you realize, oh my gosh, like, this technology is wild, that's what kind of keeps you going.
Yeah, for sure. All right, well, maybe let's pour some samples, and then we can, I think, talk a little bit more about all of the different products that you're, I don't even know, manufacturing, breeding, probably is the best term for it. And what the intention is with these, because I know you've got a range of different products, and we've talked a little bit about diacetyl and hops, and but also low alcohol, no alcohol, and that kind of thing. So shall I get some get the glasses?
Yeah, that'd be awesome.
All right, cool. So we're back. You've poured 3 samples. So what have we got here in these 3 glasses? Okay. Maybe first of all, just explain. We've got a brewery here next to us. This is a miniature brewery.
Yes,
right?
Exactly.
So you can, I presume, run the same wort and then modify whatever yeast strain is going into it, or modified yeast that you've created, to observe how it affects the outcome organoleptically.
Exactly.
And I guess in terms of foam and stability and many other things, right?
Yes, 100%. Yeah. So brewery is off camera there, where we actually prepare the wort, but we're looking at it right now.
Yep.
And then we can run that wort into each of these fermenters. We can fill up, we have 18 fermenters that we're looking at right here. We can fill up all 18 of them off of that brew house right there. But what we'll typically do is usually our experiments are 3 to 6 tanks. And so let's just say that it's 6 tanks. They're fermenting the exact same wort all side by side. So it's sort of like a well-controlled experiment. And we can vary either the genotype or we can vary other conditions. Like in this particular setup, I think we're looking at one of the things that we do with our strains once they sort of getting towards the getting closer to commercialization is we test them. We want to test them every which way and like make sure they're battle tested and ready for action. And then the other thing we'll look at is which conditions provide the best sort of conditions for those yeasts to do awesome things.
Yeah, whatever you were trying to achieve with the yeast. So then you can advise customers on temperature and time,
Yes,
pH, that kind of thing.
exactly.
Yeah.
Yeah. And like we were talking about the essentiality of nitrogen in fermentation. So the actually the second glass that you're going to taste has we were interested in how the quality of the flavor and aroma tracks with nitrogen addition. So that one has a little bit more nitrogen than the one next door to it, and we think it's a little brighter and juicier. So that's an example of as we get closer. So it starts with, hey, is this a good product? Is it doing the right thing? And we might do a lot of iteration on that. And then once we're like, yeah, this is a good product, we want to understand the conditions with that brewers can use it in to the greatest effect, basically. Okay.
Okay, so what is the difference between the three then?
Let me talk. So I'll talk about the first two first.
Yeah.
And the third one is something completely different.
Okay.
But so the first two are both sort of towards the end of fermentation. So they might taste a little bit young, if you will. And what we're looking at is a relatively, these are both strains with an ancestry that lends itself to making hazy IPAs. There's obviously a thick haze on these.
Yep.
And the parent should just smell kind of like beer flavored beer. And
that was the least technical thing you've said so far.
There you go. Yeah.
Beer flavored beer.
Just beer flavored beer. And then this next one is the exact same thing, except we've taken a very small piece of DNA.
Oh my God, that definitely smells different.
Isn't that crazy?
Yeah.
So just a tiny little piece of DNA inserted into the genome, and all of a sudden, this yeast is producing a completely different aromatic, right? What you usually hear customers say in terms of how they characterize that aroma is passion fruit is a big one. Some people get pineapple, some people get grapefruit, some people get guava.
Yeah, it's definitely tropical.
It's very tropical fruit forward, and you know
this one is, I would say, more malty.
Yeah.
This one has, a lot more tropical. Yeah.
And it's the exact like. It's on the one hand, you have a yeast that has been domesticated and brewers have been using it for decades and decades. And the other one, we just literally took a little piece of DNA and popped it into the genome. It codes for an enzyme. This enzyme creates the same chemical compounds that you would have in a passion fruit or a grapefruit. It's doing the exact same biochemistry that happens in the fruit. Or in some hops have a lot of sort of that tropical fruit flavor and aroma.
The mechanism of building like esters like this is the same whether it's a piece of fruit or it's a fermentation. You just 100%. Need the right. You need the right DNA in those in the yeast in order to make it.
Yeah, exactly. So we've basically taken a little bit of the chemistry that happens inside of a plant like a passion fruit and put it into the yeast, and now the yeast does it. So this is our kind of getting at what the various different applications are. I mentioned the low diacetyl. That's a big part of our sort of product portfolio.
Yeah.
And this strain is called Tropics, hence the sort of tropical fruit or flavor and aroma that you get off of it. And
there's no hops.
no hops in either of those. Yeah. And but what we see, again, the way we recommend using tropics is with hops, right? The thing that's cool about it, and the inspiration, I should just go back. So to go all the way back to when we started the company, we said, let's just get yeast to make hops flavor. Let's go, and we're all, ivory tower scientists at the time.
We're playing God. We can do anything.
And when you're a scientist, what you look at is you look at what other papers, other scientists have published around it. And when we read about, okay, what gives hops their hoppy flavor, we saw these compounds that are terpene alcohols in the literature a lot, like linalool, geraniol, citronellol. And we said, okay, the literature thinks that these are, hops flavor. And there's definitely a lot of truth to that, because when we got the yeast to make those chemical compounds, it definitely smells hoppy. But so then, you kind of start interacting with the industry and understanding the value proposition that brewers are, more and more. And they're basically saying, well, these flavors do smell and taste like hops, but it might not necessarily be my favorite part of hops. Okay. And so we said, all right, so what is it about hops that you like and can't normally get? And I don't know, when we started building this many years ago now, tropical fruit flavor and aroma was really big in making hazy IPAs, and I think it still is today. So we said, okay, why don't we target some of those tropical fruit aroma flavor? And so it's literally, our products are inspired by brewers.
Whatever the brewers are looking for.
And the brewers are looking for what the bartender and what the folks at the bar are looking for.
What about the bitterness of hops? I mean, my understanding is that hops are originally used as preservative as well as aromatic qualities.
Yeah.
And that, IPAs, obviously, there's much more of that kind of typically or historically that aromatic, sometimes tropical note to it.
Yeah.
Whereas some of the hopped beers from Germany, for example, like Bohemian hop beers, have less of that aromatic quality. It's more like a bitterness and a sort of woody kind of note to it.
Yeah.
But anyway, are you able to get the yeast to generate this bitterness as well as the aromatic qualities?
Yes. But again, like
are the two one and the same, or do you need more genetic coding to take place in order to make the bitterness outside of the aromatic?
Yes. So the answer to that is kind of yes and yes. There's a little bit of nuance that I can explain. So there are chemical compounds in hops that are very famous for giving rise to bitterness. And you can certainly engineer yeast to make those exact. Having said that, there are lots of different chemical compounds in the natural world that affect bitterness, right? So we even find that some of the terpene alcohols are perceived as more or less bitter, even though if you're, a student of brewing science, you would say, oh, it's these alpha acids, there's a completely different class of chemical compounds that give bitterness to beer. So the answer is you can engineer yeast to make those same ones that give beer its bitterness through those alpha acids, or you can engineer yeast to make any number of different compounds that give rise to bitterness.
Bitterness is a really weird tast sensation. It's very complicated.
It is, and nobody appreciates. It's like
sweetness, relatively simple, right? It's just is it sweet? Is it sweeter? Is it less sweet?
Yeah.
Bitterness. I mean, there's so many different compounds that trigger the sensation of bitterness, and a lot of the physiology of the tongue is really geared up for detecting, because of course in the natural world bitterness is often associated with poison, right? So you really want to make damn sure, is this thing going to poison you before it goes in your mouth. Sweetness, acidity, salt is just more about nutritional value, right? Whereas bitterness, don't. So that's really interesting that I guess these things work in concert with one another. Alpha acids and aromatic compounds in order to create a shape of bitterness. And indeed, you can engineer all of them.
And exactly.
So what's the third one?
The
Should
third
we
one
go
is
to the third,
something
or-
completely different, and what that is so basically what we were talking about earlier, the same way that you can make more good flavor or less bad, more good flavor, less bad flavor, you can also do just less ethanol. Okay. So what this is this should taste a lot like a beer, like a pretty clean kind of like a lager style
Yeah,
beer.
Pilsnery, yeah.
Yeah. So Pilsner style beer. But believe it or not, that has, I don't know, 0.3% ABV. So
Yeah,
like
it smells
less
sort
than
of malty and quite, it's got that kind of nutty Pilsnery thing going on, yeah.
awesome. Yeah, I'm pretty happy with how these beers are turning out right now. So a little bit about that. I mean, the way we think about what is possible with engineering yeast is there are all sorts of crazy processes that brewers and winemakers and fermentation folks have to do in order to control outcomes, right? And so the most drastic and what you can do with bioengineering is basically make less trouble for them, like reduce some of those process complexities that give them headaches or cost. So let me kind of contextualize that with making non-alc beer. So the way that most non-alcoholic beer is made is roughly speaking, you make normal strength beer and then you put it through a mechanical process where you separate the ethanol from the beer.
Do they distill it out?
Vacuum distillation is the most
so they'd have to heat
that's
it.
the most common way of taking the ethanol out. Yeah.
Yeah, so you're not denaturing it too much by heating it, but nevertheless, you're kind of evaporating this ethanol out.
Yes,
Yeah.
exactly. And then not only you get the ethanol out, but oftentimes you're getting a lot of the aromatics
Other volatile.
out. So then if you want to make something that's authentically beer, you end up having to add flavor back to it. So if you look at any of the biggest non-alc beers, you see that they're made with natural flavors. Right? So it's a way more engineered process. And I think the thing, and I mentioned it at the beginning that I got into engineering biology because I was passionate about sustainability implications of making biofuels and like carbon neutral fuels. And so I've always been interested in, hey, is there anything that we can do to make the brewing process more efficient? So less resources, less. And so one of the things that I'm the most excited about this technology is instead of having it, vacuum distillation is very energy intensive.
I bet it is, yeah.
It's insane. It's crazy.
Sucking all the air out of an environment, it's quite costly, right?
Yeah, and yeah, exactly. Yeah. So the thing that I'm like personally passionate about is, okay, well, first of all, that's a lot of process complexity. It's a lot of natural resources go into all that process. Well, if you can just engineer the yeast to not make the ethanol or nearly as much ethanol in the first place, like that's a much more sustainable process. It's a lot more kind of like democratic process because you don't have to spend tens of millions of dollars to install So
anyone can start brewing like that.
Yeah,
immediately.
exactly.
Yeah, and it's a one-step process.
And
right?
it's like brewing.
Yeah,
it's just like making beer. So it's a much more sort of like natural and simple process.
Although I guess there's a lot of engineering that goes into it. It's just on the front end rather than the back end.
Yes, exactly.
But you're right. I mean, you're just dealing with yeast, albeit genetically modified yeast, but you're not got all this, like you say, mechanical, and then adding flavors in afterwards to compensate for what you've taken out. That seems a bit sad.
Yeah. And of course, you can just tweak the knobs of the yeast to make sure that you turn down the ethanol production if you want to. If that, you can turn up certain flavor compounds to give it more authentic.
Yeah,
like,
I mean, you can combine all these
yeast
things together, right? So you can do a low ABV, like nearly zero ABV ferment, but then also go, right, I want it to have IPA-like qualities and this level of bitterness and so on. And
yes,
yeah, that's just amazing.
Yeah. So that's something that I've been, that's an application area that's kind of near and dear to my heart.
So you've got breweries out in the wild that are using this kind of yeast to make their non-alc beers, low alcohol beers.
Yeah. And one other thing on that note, we really started the business on flavor, more and less, more good flavor, less bad flavor, and then we started branching into other applications like less ethanol. But I think the potential is there also not is also in not just flavor and ethanol, but other compounds that affect like the healthfulness of the beer that you are drinking. So like these are the kinds of applications that I'm also very excited about, is if you can make beer. I mean, beer has always been consumed as like, well, maybe not always, but there's definitely points in history where beer was the healthiest option. You know?
I mean, it's only really the last 20 or 30 years where people have decided it's not so healthy. But yeah, outside of that, we've had 10,000 years of beer being the health option. Because they say you got to boil the water so that sterilizes it. The alcohol content of it keeps it safer longer than a pail of water is going to stay safe, or whatever other. That milk's gone sour, you know.
Yeah.
So yeah,
and you know, nutrition. You know, there's all kinds of crazy history of how armies were, you know, fueled by beer, and it was the best source of nutrition.
Well, Guinness is maybe the best example.
Yeah,
right? I mean, used to be given to pregnant women. I think up until 2009, if you went and gave blood in Ireland, they gave you a pint of Guinness afterwards just to top your iron levels up. That only stopped 16 years ago.
Yeah.
That literally nurses were handing you a Guinness, sort of like, there you go, that'll make you feel better, you know?
Yeah.
But yeah, maybe we need a little bit more of that. So what can you do to make a glass of beer healthier? I mean, besides, I guess, taking the alcohol out, what?
Well, that's a good starting point. There are other chemical compounds in beer that are like that may have adverse health impact. So you can take less, you know, you can take some of those out. And then there are chemical compounds that are good for you that you could consider, you know, potentially put in there. So, you know, you really are going back to, you know, you could really go back to that world where, well, maybe it's a non-out beer that you're giving the pregnant woman after, or giving the blood donor after they donate, but maybe that is how you top off in one.
So what sort of compounds are you talking about here? Like, is it like iron, or what are you?
Well, like okay. So I don't want to, without getting, you know, too specific about what we're working on. Let me just give you an example of, like, you know, you eat broccoli because you want to. So maybe people don't actually generally appreciate this, but eating broccoli can prevent cancer. And that's because there are certain chemical compounds in broccoli that have cancer-fighting properties, okay? So you can imagine a world where you're engineering chemical compounds that prevent cancer. But you could also imagine things that are just like, well, that has a quality, like just general cell health, right? Like, oh, these chemical compounds are really important for promoting general cell health. So stuff like that is also kind of fair game.
This is wild. So, in the not too distant future, beer could be a legit health drink.
Yeah, and I don't know how. Like, I'm not going to say it's going to be next year, but I think certainly in the next decade, that is going to be. It is a great mechanism for delivering.
Well, you've got to have the fermentation component as well, right?
Yes.
So it has to be something that is fermented. So you're looking at beer, wine, cider, and other fermented beverages.
Yeah.
Kombucha.
Yeah.
Kind of thing.
Absolutely.
Yeah, because you can modify the genome of you know, that SCOBY, as isn't it, symbiotic culture of yeast and bacteria.
100%.
Yeah. Wow. Okay.
So I mean, all right, we used to have this old tagline, which we've moved away from, but the tagline was like, the possibilities are endless. And that's, it is, yeah, it's hard to kind of imagine the boundaries of what is possible with bioengineering. But we can promise that the beverages are going to be generally better and generally more healthful in the future.
And tasty.
Yeah. Sorry, better. I meant more delicious. More delicious, more healthy, both.
It's wild. Okay, so Are you only working with beer at the moment, or are you involved?
We're also interested in wine.
Yeah.
I mean, we're interested in just about anything, but we're actively developing products for the wine market as well.
Yeah.
And so again, more good flavor, less bad flavor has been our initial focus areas.
And wine really needs some help with non-alc. I feel as
Yeah,
well. There's
yeah.
really very few good. I was talking to a guy who's he was a product developer. He's in his 80s now, but he's called David Gluckman, British guy. He came up with a lot of spirit products that you'll know. Bailey's was his invention.
Oh wow.
Smirnoff Black, Tanqueray 10, Ciroc vodka.
Oof.
He came up with the idea of using grapes. So real sort of like product guy. He's not a technical guy at all. He just comes up with the idea. And his theory was that non-alc beer works really well because the difference between 4.5% and 0% is pretty small. Whereas with wine, you're taking away a significant chunk of what is in that bottle because wine's, 12, 13, 14% ABV. But now I'm thinking, can you get the yeast to generate taste properties that will simulate alcohol, the burn or whatever, trigeminal thing is going on there?
Yeah.
Are there compounds that can be introduced that aren't ethanol, but behave in a similar way to ethanol that will then simulate the same thing as when you take a sip of wine, you're like, wine doesn't burn, but it has that tactile effect of, there's booze in there. And in addition to that, of course, like the formation of the esters that you'd expect to have in a nice glass of wine.
Yeah.
So
of course.
Yeah.
Yeah. I think there are intermediate steps towards that kind of, developing that technology is not straightforward because you have to make up for a lot of stuff that
exactly
doing.
Yeah.
But I think where I see the application areas being in the next few years is, well, first of all, if you can go from a 15% ABV wine down to 10, let's just say 10, and still have it like present with that authentic wine character, I think that there's a big market for that. I also know that, and again, my brain always goes to like the climate related stuff. So I'm one of the things that you observe in Napa Valley, which is my point of reference because it's only a few miles up the road, is the grapes are just getting more and more sugar in them, which means when you ferment them, they're just higher and higher ABV products as the summers get hotter, etc. So if we can, the same sort of use of that application, if you can dial it down from 15, 16 down 10, I think there's some really, really interesting opportunities there. I think if you can dial it down to 5, there's yet another like.
Well, wouldn't it be great to be able to drink 3 times as much wine? And
Yeah,
have the same
exactly.
alcohol effect?
Right. And I think it will also affect the occasions where wine is being consumed, right? Because right now it's like a bottle and it's, between 2 people at dinner, let's just say. But if you have these beautiful 5% wines, maybe now you're looking at like something that's sessionable. You can drink a few.
On draft in a pub.
Right. Yeah. Right.
Glass.
Yeah. Exactly.
You're right. I mean,
sit with your buddies and a couple of them.
I mean, the knock-on effect, like you say, to sort of social environments is hard to kind of. Well, you can't really predict how it might affect these occasions and create new opportunities for drinking wine, enjoying wine, different places. Yeah, it's wild. Here's a question that just occurred to me. If you're creating a non-alcoholic or even a lower alcoholic product, what happens to the sugar that's not turned into ethanol? Where does it go? What is it turned into instead?
Yeah. So in the case of beer, there is a significant amount of maltose in the finished product. It just doesn't get fermented at all. And it's funny when I set out on this journey. All right. So we developed these products only in the last couple of years. Part of the reason, and I had this, and now I'm going to expose like how naive I was and like how bad I was at food science. But I assumed that part of what gave bad non-alcoholic beer its sort of okay. When people beat up on non-alcoholic beer of yesterday, they describe it as sweet, cloying, worty. Like
Yes.
those are the three things,
Yes.
right? And I just assumed that that was due to residual maltose. But it turns out that maltose, that it's a completely different set of chemical compounds that gives all that like wordy, cloying sweetness. Maltose on its own is actually pretty darn inert, believe it or not. In fact,
It's
if
not a
anything,
very sweet sugar. Yeah.
no, it does have an impact on the body of the product, but that's almost a good thing to have like a little bit of extra maltose to
Yeah.
kind of bulk up on that. Yeah.
Mm.
Yeah.
Yeah, because that doesn't taste sweet.
Yeah. Right.
It's not sweet tasting. No.
Yeah.
No.
Yeah.
My background is more spirits than beer or wine. So, from a purely selfish point of view, what opportunity is there in the world of spirits? Because, fermentation is still critical to the production of all spirits. And having been to quite a few distilleries and spoken to a lot of distillers, I'm well aware that some of them are experimenting with yeasts. They're trying to engender certain aromatic qualities into, let's say, whiskey, for example, because it sort of sticks with beer.
Yeah.
They're trying to brew the beer, the wash that goes into their stills, to have certain esters, compounds in there that give certain aromatic qualities. The idea being that when it's distilled, they can concentrate those, capture them, and they in turn will affect the whiskey as it matures, and all the chemical reactions that go on inside a cask as well. Yeah. So there's opportunity there, is everyone getting at, to do this same kind of work for the spirits industry. I mean, it sounds like you've got a lot on your plate as it is already, but is this something long term that you?
Oh, absolutely. Yeah, yeah. I think there's a lot of opportunity for more good flavor in spirits, and efficiencies. Again, this is something that I'm really kind of passionate about. Is identify, like, if you can make the process of fermentation more efficient, that can also kind of.
Yeah, there's so much. I mean, as a product, then it becomes financially very desirable, right? Yeah.
Yeah, that is the cool thing about a lot of these. When the economics, like, I think when you think about how can you make the world more sustainable, I think one of the things that became really obvious to me early on is that nobody's going to pay a premium just for something to be more sustainable, or at least the vast majority of folks are not going to. So finding the places where the thing that's more sustainable is also more cost effective, those are the technologies that are going to lead to sustainability with the least friction.
Cost efficiency, in a way, is sustainability as well, right? Exactly. I mean, the two are not disconnected from one another.
Exactly. Yeah. Like, if you have to buy, I mean, in the case of vacuum distillation and non-alcoholic beer, if you have to pay more money to buy the energy, then that's going to be deterrent enough, and then you're shifting. And then if you decide, oh, okay, I'm going to shift away from that to a greener, to a more cost-effective way of making non-alcoholic beer. Oh, and by the way, it's way more sustainable. It's like, those are the technologies that I want to be working on.
Yeah, for sure. Right, so practical application of this. I'm a brewer, let's say.
Yeah.
What happens? You just turn up with I pay you some money, you turn up with a bag of yeast, and then we never see each other again? Or do I need to keep buying it from you? Have you engineered these yeasts so that they have only a certain lifespan, and then I've got to be like a subscription customer? Is this how?
Typically, for regular beer, we engineer it to be the yeast strains to be hardy.
Yeah.
But we do ask our customers to buy at some cadence. Like, don't just like take the yeast strain, put it in a bank, and like grow it up yourself. Like, give us some recurring sort of business, and in exchange, we'll provide you with the very best quality of yeast. It will be completely pure of contaminants, and so if I'm a brewer, again, I'm trying to create technology that you want to kind of go with the flow, right? So here's how brewers tend, at least most of our customers, the way they tend to treat yeast is they say, okay, I'll buy liquid yeast slurry from you in a bladder, and we'll ship it to them in a cardboard box, and then they'll pitch that into one tank. It'll make great beer, and then they'll repitch it and repitch it and repitch it. And then the further you repitch it, there's a bunch of reasons why you would want to go back to the well. The first reason is the genetics are going to drift, something we've talked about already. You can, in fact, a lot of brewers will say, yeah, every brewer that has done a lot of serial repitching will say, yeah, this strain was clear, and on the 3rd generation or the 7th generation, all of a sudden my beer was all hazy. Okay, so there's that like genetic drift piece. And then there's also, like, as you repitch and repitch, unless you're brewing beer in a clean room that's like perfectly sanitary, you're going to start introducing. Now, the yeast is good at kind of warding off some of those competitors, but still the pitch is going to get less and less pure. Mm-hmm. So it makes sense for them to kind of come back to the well, and then we resupply. So that's thing number one. The thing about non-alc is that that is what brewers call a pitch and ditch application. And the reason why is because their purity of the yeast is even that much more important, right? Because you don't have ethanol, the preservative, to fight off all the, so you can't safely repitch from tank to tank.
Yeah, got you.
So that is kind of a downside of these non-alc yeast products. But it's something that, we gotta, like, we're safe. We're a safety first company. That's thing number one, keeping the consumer safe.
Yeah, I mean, I guess, and besides bacterial infection, you could just get wild yeast that starts making ethanol as well.
Yeah. That will
also
happen.
Not good.
Yeah, that will totally happen. So, we're really gung ho on that. And the one other thing that I should mention is that we kind of talked about democratizing non-alc brewing. The one way in which it is not as easy as making beer is that generally the best safety practice is to put the beer in cans and run it through a tunnel pasteurizer. That is the gold standard for safety. That's what we encourage our customers to do. So if you don't have to have a tunnel pasteurizer, that becomes hard. But we're also very interested in technologies that can make tunnel pasteurization easier and cheaper for folks to bring in-house at smaller scales.
Just amazing. So if I ordered yeast from you, it's what, FedEx to me kind of thing?
Yeah. We only ship in the US at this point, but soon we'll be sending to the UK and other.
Oh, cool. That's good. That would be great for the brewers of the UK.
Yeah.
You got much competition in this marketplace?
There are a couple other companies that are modifying yeast for the brewing application.
Are they based out in this direction as well?
The U.S. market is the friendliest to these products, so there are and I know that there are other countries that are also pretty friendly, and as far as I understand it, the UK is pretty friendly. Canada is pretty friendly, and then some of the South American countries are pretty friendly as well. I know that the general the conventional wisdom is that EU countries can be a little bit more resistant.
Yeah, so how does that work? Like the sort of stigma of genetic modification. What are you doing? Are you doing anything in terms of sort of education besides obviously podcasts?
Yeah,
to talk about this.
I mean, it's so funny in the U.S. All right, let me go back to when I'm wearing a lab coat in the bioengineering lab with Jay Kiesling, this pioneer of metabolic engineering, and he's done a gazillion startups, and including, he did a startup around yeast that produced this anti-malarial and biofuels and bioplastics, you name it. And I said, hey Jay, like, what do you think about this idea? And he was basically like, I think it's a really good idea. The only question that I have is, bioengineering in food or GMO in food. There's just a lot of baggage there. And then and then I went to this brewing science professor, Charlie, and I was like, Charlie, what do you think about this idea? And And he was like, Charles, I think you're absolutely nuts for trying it. It's never going to go anywhere because, and trust me, because I tried back in the 90s. We had, a university, developed these strains, and they had some beneficial trait, but nobody wanted to touch it because of the GMO stigma. So that was when I was starting the company, I believed that that was the number one sort of dragon that we would have to slay.
Yeah.
And now 8 years later, it's in the US, it appears to be a complete non-issue. Like, we have hundreds of customers. And I remember, and it's just it's one of those things where back in, let's just say 2020 was when we really started, 2021 is when we really started selling an appreciable amount of liquid yeast to brewers. And back then, like, it was kind of on, everyone was really excited about it, but it was also kind of on everyone's mind. It was like, I feel a little bit like of hesitation around it. And I don't know how my consumers are going to respond to it. But what we've seen over the last 5 years is, and it's like one of those things with technology where you're like, well, it probably won't be that different next year, but 5 years it's going to be like completely different. And sure enough, that's been what we've seen with the stigma around GMO. And in the early days, I remember we were working with small brewers around here, and they would message it. They wanted to be, that was the cool thing about our early adopters were all small craft breweries, and they wanted to be fully transparent about it and message, hey, we're using this new technology. It's safe, and we feel like it's going to elevate beer. Okay. And that's why we're giving it to you. It's not to, and this is kind of going back to why GMOs maybe had such a negative stigma against them in the first place. Obviously, there's something, like, I'm not trying to minimize just like messing around with my food. Like, that's intuitive to me too. It's like, I want food to feel natural and be natural. But another piece of it has always been like, well, is a safety component. So one of the things that we have done is we've gone kind of to the end of the, like, we go an extra mile and then like an extra 10 miles to ensure the safety of these products. And the way that we do that is we measure, we have in the other room over there, we have a GCMS and an LCMS, and we can quantify the levels of thousands of different chemical compounds that are in the finished beer. And so what we do is we ensure when we engineer yeast to make some of that tropical flavor and aroma, that only those flavor and aroma compounds are the ones that are changing. Thousands of others are not changing. And the only reason we would engineer the yeast to make those things in the first place is if we know that they're safe. And a big part of that is, hey, passion fruit, like, it's the same chemical compounds that are in passion fruit. Passion fruit's been eaten for only a long time, and it's proven history of safety.
Yeah, never heard of any issues with passion fruit.
Yeah. So exactly. So, like, to come back to, that was the number one concern of, sort of like existential concern around whether this technology would take hold. And years later, it's like, at least in the U.S. market, the biggest. Okay, so I sort of lost my train of thought. I'll try and break. The biggest thing for brewers was understanding that consumers would not have an adverse feeling about it. And it literally took dozens of breweries being very transparent and seeing that consumers were just enthusiastic about better beer. The final thing I'll say about it is that the GMOs of 40 years ago were all, the benefits were going to the farmers. It was all about them being able to save $1 less, whereas today it's like, well, we're engineering traits that either make the quality of beer better or make the brewing process more sustainable. Like, there are actually benefits that are important to the consumer. There's a reason why, we're elevating beer.
Yeah. No, I get all of that. I think, and a couple of points. I think a lot of the problem with the public perception of GMO was that it felt like there was some kind of economic advantage that was being exploited out of people. Higher yields, bigger crop, and oftentimes at the forfeit of flavor as well, and nutrition.
Yes.
And the thing is, it doesn't need to be that way. It can be the absolute opposite way around, right?
Exactly.
You can, get a more flavors in tomato or more nutritionally dense corn or whatever it is.
Exactly.
And so it's just throwing the baby out with the bathwater if you just say, oh, GMOs, no, I'm never going to eat a GMO ingredient. And also going back to what we were talking about near the start, where it's like everything we eat's been genetically selected, if not modified. So it reminds me a little bit of like microwaves. Like in the UK anyway, microwaves have this stigma. Oh, you got a microwave. Oh, what are you doing with your microwave? And it's sort of seen as this unhealthy device, probably partly because people don't really understand how they work.
Yeah.
And microwaves can be unhealthy because a lot of people put shitty microwave food in there. But like, you don't have to put shitty microwave food in your microwave. You can steam vegetables in there,
Yeah.
So I think people are coming around, and understanding more and more that GMO can be super beneficial. And like, you're going back to what you've talked about sustainability, as with climate change, if we can modify rice, for example, to be more tolerant to heat or drought or whatever it is in parts of the world where they really need food, like, it's pretty unethically sound to not do that, right? What, because we have sort of an ickiness about genetic modification? Fuck off.
I completely agree.
So, yeah.
You can literally save, like, there's these, there's such amazing examples how you can save people's lives by engineering, additional nutrition into rice, for example. And you got people, oh, it's a GMO, and tearing the rice, the experimental rice, and you're literally saving, I'm trying to recollect the details of this set of trials where scientists had, I think it's like I want to say it's vitamin B12, but some vitamin that leads to like blindness in malnourished individuals. And so some well-intentioned group of scientists were like, okay, well, we're engineering, vitamin B12 into this rice. So then everyone that eats the rice, nobody's going to get childhood blindness anymore. And there's people, protesters, and jumping over the fence into pulling the crop out. And you're like, really? It's such a well-intentioned.
People don't like it. I mean, like, it's like fluoride in tap water and that kind of thing. And I mean, I get it. Like, if you don't have a choice, then I can see why some people get upset. And I mean, like, let's not get into the whole vaccine thing here because that's way off topic. But like, there is probably a line there at some point where it's like, this is your government mandated set of minerals that we have modified into every single plants that there ought to still be the option to opt in or out.
Yeah.
But then again, if you're in a part of the world where there's some, malnutritional disease that is afflicting a large portion of the population, along with famine and whatever else, and you have the opportunity to make a really big difference, then I think ethically you have a responsibility to do that, but there's always gonna be someone that's gonna fucking disagree, right?
Well, I think like, I think what you're getting at is that a commonality of technology is that it can generally it can be used for stuff that's good for humans, and it can be used for stuff that's bad for humans. And I think that genetic engineering is, I mean, like computers, good stuff and bad stuff, right?
That's certainly true,
right? GMO, genetic engineering, good stuff, and and bad stuff. And the same thing is true with I mean, just thinking about the vaccine thing, pure, like in a very sort of theoretical way, like there are ways that vaccines can deliver bad stuff, and there are ways that I guarantee vaccines can deliver good stuff. Like this decade, we're going to see cancer cured by vaccines. Now I'm sort of going out on a limb here. That's my big prediction.
You went there
for 2020 or 2030. That's what I'm saying.
That's going to be the title of this episode.
Yeah, vaccines cure cancer. Oh no.
I'm going to turn it into a health episode. Just cut out all the beer nonsense at the start.
Whack job scientist studying yeast in beer thinks that he's going to cure cancer with vaccines. No, I think it's going to happen.
Just a final point on the GMO thing. I mean, with beer and brewing, it's an interesting one because there's sort of a degree of separation there in that yeast isn't. I wouldn't class yeast as an ingredient.
Mm-hmm.
I'd class it as a organic mechanism.
Yeah, like a processing aid is often
and so you're obviously modifying the yeast, but not necessarily the barley. I mean, that could be modified too, of course.
Yeah.
So there's a sort of half a degree of separation there in a way.
Yeah.
Like the product you're drinking, I mean, some beers are yeasty, but a lot of the time the yeast is completely removed out, right? And so that part is not in it anymore.
The beer is not GMO. We use the processing aid is a GM. Okay, so the process does rely on genetic engineering, but the beer itself is not a GMO.
Yeah. Okay, so I mean, you've already talked about what the future holds in terms of your next steps and ambitions, and it's remarkable, and obviously the cure for cancer.
Yeah, of course.
Great. Yeah. I mean, thanks. This has been absolutely amazing.
Yeah, no, it's been a really fun conversation.
Charles Denby, thanks so much.
Cheers. Thank you.